Hi, I'm Mr. Enzert, and today we're going to talk about one of my favorite topics, Mendelian genetics. So we'll start with why we call it Mendelian genetics. It all started in the 1800s with an Augustinian monk named Gregor Mendel. And prior to Mendel's experiments, a lot of people really believed that inheritance involved a blending model that two parents' traits would kind of blend together into their offspring. So, an example of this would be like a really tall parent who had kids with a really short parent. So, their kids should have a height somewhere in between. Or another example is somebody with a really big nose had kids with somebody with a really little nose. Well, their kids should have a nose that was right in the middle of those two. That would be the blending model. Now, we know the blending model doesn't actually work. If it did, over lots and lots of generations, eventually we would all look exactly the same because we'd all work towards an intermediate form of every trait. And that doesn't happen. That's not how genetics happens. So Gregor Mendel is considered the father of genetics because he came up with his understanding of inheritance through experiments with peas. So as he worked in this monastery teaching math and science, he started tending the garden of this monastery. And one thing that he noticed about the garden, there were pea plants growing in the garden, and he noticed that some of the plants had white flowers and some of the plants had purple flowers. And so he was curious to see what would happen if he could cross those two plants. Now, in nature, we know insects do this work. So what Mendel did was he took a tiny little brush and he took some of the pollen out of one plant and he painted it onto the sex parts of another plant. And so he was forcibly crossing these plants with the desired traits that he wanted to see. So here's Mendel's experiments. He took plants with opposing characteristics, different traits. And there's actually lots of different traits in pea plants. There's flower color, pea texture, the shape of the pod, how tall the plant is. So lots of different traits that he was looking at. We're just going to stick with the flower color for this example. What Mendel noticed was when he crossed a purple flower with a white flower, all of the first offspring were purple. So that all the seeds produced grew into plants that produced purple flowers. So that was kind of interesting. And then he did something, took it a little further, and he took that second generation, those purple plants, and he crossed them with each other. And what he noticed, something kind of intriguing, what he noticed is that about 25% of the time, a plant would be produced with white flowers. So what this told Mendel was that somewhere in that plant, those purple plants from the second generation, somewhere in them were the instructions for making white flowers. They were just hidden. So I want to talk about purebred just real quickly. This is a word we use a lot. People talk about purebred dogs or other animals, but the word purebred or pure breeding just means that plant or animal will always produce a specific trait that you're looking at because that's the only version of alleles that plant or animal has. So if it's pure breeding, for example, a purple flower, those purple flowers, when crossed with other purple flowers, will always produce purple flowered plants, and the same with the white. So that's what purebred means. It just means that the plant only has one version of the genes for that particular trait. So that leads us to what is a gene? So a gene is a piece of our DNA that codes for traits. And there's a difference or a distinction between gene and allele. An allele is a gene, but an allele is specifically a particular type of gene. So when you have multiple types of genes possible, so for eye color as an example, brown eye color and blue eye color, those different versions of that gene are called alleles. So we have a brown allele or brown version and a blue allele, the blue version. So gene is the bit of DNA that codes for a trait, and allele is the specific version of that gene that you have. All right, so in humans, we have more than 20,000 genes, and we actually have 2 copies of each of those genes. We get one from our mom and one from our dad, and so most of the time, that's the case. There are some rare genetic disorders where it's a little different, but most of the time, every single one of us has 2 copies of each of these 20,000 plus genes. And our genes are packaged on strands of our DNA called chromosomes. And so on each chromosome, we have many genes, and we receive 1 copy of each chromosome from each parent through the process of meiosis. Random shuffling of their chromosomes gives us a random assortment of their genes. And that's how we get the genetic variation that we get from our parents. So I want to talk about Mendel's experiments and what he determined from his flower cross. So he noticed that after crossing the purple and the white flower, all of the first generation were purple. And so Mendel realized that some traits must be dominant. And a dominant trait is one that overshadows or hides the other version, or the recessive trait. So Mendel came up with this law of dominance. That's one factor in a pair of traits dominates the other in inheritance, unless both factors in a pair are recessive. So in this case, we're simplifying eye color here. But if you have 2 of the big B alleles, then you have brown eyes. But if you have 1 of the big B alleles and 1 of the little b alleles, you still have brown eyes because brown is dominant. And the other example, the blue-eyed trait, the only way to get blue eyes is to have 2 of those lowercase letters, 2 of those recessive alleles. You need to have 2 blue alleles to have blue eyes because that's the recessive trait. All right, a couple of terms that are really important to know. The first one is genotype. Genotype. It means the specific combination of alleles an organism contains for a particular trait. It's the type of genes you have. And there's 2 ways to have those or name those. Homozygous. Homozygous means you have 2 copies of the same allele. So for this purple plant here, a big B and a big B makes a purple plant. And a little b and a little b makes a white plant. So those plants are purebred. They only have 1 copy, 1 type of allele. They just have it twice. And the other name for genotype is heterozygous. Hetero means different. And heterozygous plants have 2 different alleles, 1 dominant and 1 recessive. We also call these plants hybrids because they have 1 of each. And the other term that's really important is phenotype. So we had genotype, and now we're talking about phenotype. Phenotype is the physical or observable characteristic of an organism. So ultimately, our genotype or our genes determine what our phenotypes look like. Genotypes code for our phenotype. So all of your genes make you look the way you do because they produce the factors that give you your traits. So genotype is your genes, and phenotype is the physical or observable characteristic that you have. One other thing, before we get into an example of Punnett squares, I just want to talk about reproductive generations. So when we're doing genetic crosses specifically, it's important to be able to name the generations. The first generation is the P generation. That's the parental generation. And most of the time, this is actually going to be a cross between purebred individuals that have 2 of the same alleles. After you cross P generation, you get what's known as the F1, or the first filial. That's the first offspring from that cross. And we could take that a step further and cross those F1 offspring to produce an F2, and so on and so forth. Generally, in these genetics problems, we don't go beyond F2 because it starts to get really complicated, really convoluted, knowing which organisms to cross. So we're just going to stick with this, P, F1, and F2. So a good example of this would be like if you were the F2 generation, your parents would be the F1 generation, and your grandparents would be the P generation, just working back in those generations. Okay, we're going to get into a cross, an example. So I'm going to read the question. What are the expected outcomes of a cross between a purebred purple-flowered pea plant and a recessive white-flowered pea plant? So a purebred purple-flowered plant, if it's purebred, that means that it has 2 of the same allele. So I'm going to draw the first plant's genotype on top of this Punnett square. So we're going to draw a big P and a big P, and that represents this guy, this purple flower. Okay, I'm going to put the white flower genotype on the side, and so that's going to be a little p and a little p. Sometimes it's hard to tell the difference between p's, so I'll just draw a little line underneath these guys. Okay, that's my lowercase p. So in order to use the Punnett square, this is just a bookkeeping tool for genes. We have to combine these letters together. So I'm going to move the big P's down into the boxes. I have a big P here and a big P here, and second big P moves down, and again it moves down. And then we're going to do the same thing for the little p's, and they're going to move across in those rows. So we have a little p, a little p, a little p, and a little p. All of the offspring in this cross have the exact same genotype. So 100% of these offspring express the dominant trait because big P, purple, is dominant. And because they all have one big P, they are all going to be purple plants. But they are carriers of that recessive trait. They all have that little p, that white color. They just don't express it because it's recessive. Okay, now we're going to go on to the next example. This is called a hybrid cross. So a hybrid cross is where we take those F1 offspring and we cross them together. So now I have two heterozygous purple flowered plants. Heterozygous, that means that they are big P, little p, so that's this guy, and big P, little p, and that's this guy. Okay, now we have to move those letters into the Punnett square. So the big P moves down, and this other big P moves across, and this big P moves, oh, oops, I'm going to undo that. This is a little p, and this is a little p, and these two guys, I should have changed colors. These two guys are little p's as well. Okay, this time I got completely different results. Okay, my results. I have when I look at genotypes, 25%, right here, 25% are homozygous dominant. Big P, big P. Okay, 50% are going to be heterozygous. Big P, little p. That happened twice. And 25% are going to be homozygous recessive. So my genotypes are different for this cross. I have 3 different genotypes. But if I want to know the phenotypes, what these plants actually look like, it's going to be different again. So if I look at the actual phenotype expression for purple, well, this one's purple, and this one's purple, and this one's purple. So 3 out of 4 of these plants are going to be purple, and 1 out of 4, or 25%, are going to be white. All right, so those are examples of Punnett squares, using Punnett squares to solve problems, and I hope this video is helpful.